here. The first C. Stephen Foster Honorary Lecture was given last year by Dr. Bartley Mondino from UCLA. And this year, the second C. Stephen Foster Honorary Lecture will be given by Peter John McDonnell from Wilmer. Dr. McDonnell was born in Paterson, New Jersey, and he earned his BA in chemistry from Dartmouth College with a summa cum laude honor for graduation. He earned his MD from Johns Hopkins and did a residency at Wilmer, and he was a chief resident, which is at Wilmer, it's a distinguished honor and was additional year. Am I correct, Dr. McDonnell? Yeah. And he did his fellowship in cornea external disease at USC in Doheny. And in 1999, he was named the Irving H. Leopold Professor and Chair of Ophthalmology at UC Irvine. And in 2003, he became the director and the William Holland Wilmer Professor of Ophthalmology at the Wilmer Ophthalmological Institute. His research interests includes dry eyes, ocular infection, and gene therapy to control cornea wound healing and inflammation. He's a recipient of numerous research grants, including ones from NIH and Research for Prevent Blindness. He has published over 270 peer-reviewed articles, and he is the editor-in-chief of Ophthalmology Times that many of us receive in our mail. He holds 8 patents, and the American Academy of Ophthalmology has honored him with the Honor Award in 1991 and the Senior Achievement Award in 2001, among the many other awards he has earned. He is a co-founder and a scientific advisor of the advisory board member of the Graybug LLC. And in 2013, he was elected president and the board of director of the National Alliance for Eye and Vision Research and the Alliance for Eye and Vision Research, which are nonprofit organizations dedicated to the education for the public as well as the Congress. And today, he is here, and the talk is on, the title is Improve the Health of Mankind and to Create a Company. Dr. McDonnell.
Well, thank you very much. It's a delight to join you here in this beautiful venue for something special, which is to be able to speak about somebody who's made a dramatic contribution, cast a giant shadow in ophthalmology. Just for disclosure, I am on the board of director of Allergan, large pharmaceutical company, and was co-founder of a company based on technology developed partly in my laboratory. So Steve Foster is someone that I've never trained with. I've never lived in Boston or did any of my training there. And he became on my radar screen largely initially because of his scholarly contributions to the field, where I think he, in addition to his 900 publications, peer-reviewed publications, his books, helped to really cement him as the leader in understanding in this country and understanding the role of inflammation in contributing to ophthalmic disease. But then I did my got my first job and did my cornea fellowship out in Los Angeles, where I worked with Ron Smith, who ran the cornea service, and Steve Ryan, who was like a special mentor and role model for me, and my first department chairman gave me a job. And I would hear this name mentioned all the time, his fellow Steve Foster, usually with tremendous admiration, sometimes interesting anecdotes. Ron Smith liked to tell a funny story every so often, and sometimes that name would appear. And I learned that there were these camps. There's the Proctor group out here in California, and then there's this group in Boston. And there was sort of a healthy interplay amongst those groups. But what really made me sit up and take notice was I would begin, as my career went along, I would meet all these people, invariably very, very successful people, and I would find that they had trained at some point in their career with Dr. Foster in Boston. So he threw this, and perhaps he might say one day as he's thinking about what's been his legacy and contribution to the field, it may be that he would agree with my assessment that it may be all the individuals whose careers he's helped launch and have become very successful academicians and clinicians around the country and the world. And the fact that his favorite works for me, his favorite trainee works for me, is, I think, a particular plus. Now, Esten is your favorite trainee, right? Isn't that correct?
Number one.
Because I hear her mention that, I would say probably weekly to me, and 3 times during the week where we do our salary reviews at Wilmer. She mentions that she stands out amongst your trainees as the best. So I'd like to tell you a story, and the story is about a young cornea academician, an assistant professor out in Los Angeles who realizes that he's seeing a lot of patients being sent his way with neurotrophic keratitis for one reason or another, after herpes zoster ophthalmicus, after surgery where the trigeminal ganglion's been impacted, whatever reason, these people having non-epithelial healing defects. And around this time, just a couple of years before that, the Nobel Prize was awarded to a group of scientists who discovered NGF, this nerve growth factor. And so this young assistant professor submits a proposal, fairly straightforward, where he wonders, gee, if there's a lack of innervation now to the cornea, maybe we could treat these people. I would treat them by sewing their eyes closed, not infrequently at this time. And I'm thinking maybe there's a better way to treat it, find out what neuropeptides and neurotransmitters, what things are missing, no longer available to the corneal epithelium. And perhaps this would allow us to better treat this group of patients more successfully. So this assistant professor submits this proposal to ask for funding, I think the princely sum of $25,000 at the time, and receives what I think is probably the most negative review in the history of a proposal dealing with eye research. And basically it says there's no merit to this idea. And I get along back with it. It doesn't say who authored it, but it has a list of people on the committee, and it's the prominent academicians of the day, department chairs and so on, all these people who, as a young trainee interested in cornea, I'd looked up to and read their papers, maybe not all of them, but most of them. And so I knew if this group felt that my idea had no merit, then it had no merit, and it was time to move on to something else. So we now go 31 years later, and what happens? What happens is that the FDA approves the first drug for the treatment of neurotrophic keratitis using recombinant nerve growth factor. So fortunately, there were much smarter, more persistent individuals that figured out, hey, there's an opportunity here and we should pursue it. So I had that experience, and then in case you didn't guess, the young assistant professor in that story, that was yours truly. But now I'll tell you another story. This is a less young associate professor, cornea specialist, and he's noting, at the time that this person trained, there weren't a lot of therapies available for glaucoma, but one of them was topical pilocarpine. You had to give it to patients 4 times a day, and they'd get headaches, and they hated it. But we felt that it was believed to work by stimulating muscle contraction in the ciliary body and increasing outflow of aqueous. And so this associate professor thinks to himself, gee, maybe there's an opportunity to do nerve stimulation using perhaps an electrical device. And he's introduced to a vice president of a company that was the leading company at the time in cardiac pacemakers, and also looking at stimulation, spinal stimulation device to control pain and certain other conditions. So this associate professor meets with this individual and explains these ideas about might it be possible to directly stimulate the ciliary body musculature, or perhaps stimulate lacrimal gland tissue in a way to promote tearing and treat dry eye disease, which was quite poorly understood at the time. And there was no, it was, well, we'll think about it and get back to you, which was code for we won't get back to you. And I guess this company had bigger fish to fry, and it was clear at the meeting they didn't think the eye business was a very attractive business. It was small and not very interesting. So the concept sort of went nowhere, and the associate professor didn't consider talking to any other companies or pursuing it further. And we're now a mere 25 years later when the FDA approves a device for stimulating, intranasally stimulating lacrimal gland secretion. And the genesis of this device was a group of undergraduates at Stanford who were rotating and looking at in ophthalmology clinics, and noticed that in just about every clinic wherever they went, it seemed about 80% of the patients had dry eye, and they thought, well, maybe there's an opportunity here and we should pursue it. And they did, and they were successful. So, what I would say is, in part, we're all children, we're all results of our upbringing. My father used to quote this phrase that I guess originated from the Greeks, which was that the child is the father of the man. So early on, you get certain exposures, and you learn as a trainee. You learn by interacting with your senior faculty, people that you really admire, who've contributed so much to the field. You see how they conduct themselves and how they think, and you maybe internalize a lot of that. I heard this a lot when I was a medical student at Johns Hopkins in the late 70s and early 80s, and that is we doctors serve the sick and we don't profit from those who have illness. And what they meant by that was we don't get involved in the industry side of things. And if you had been a faculty member at Johns Hopkins at that time and said, gee, I'm interested in patenting something and maybe working with industry, you would have been invited to leave, because that was not really something that was becoming of a Hopkins faculty member. And then I would think to myself also, well, I'm just one person. What can I do compared to these great big companies? And then what do I know about business? My own father would tell me, he was a general surgeon, and he would say there's a certain thing that is common to all physicians, and that is that they're terrible business people. And they're terrible with money, and they don't know how to do business. So I believed growing up, doctors serve patients and take care of patients, and business people, they know business, they're really good at it, and they handle the business side. And then I thought to myself, who would I be, in that experience I had as an assistant professor, to second guess the most prominent academicians who've judged that my idea has no merit, because I knew you don't rise to the top and you don't become a world famous head of a division at a place like Mass Eye and Ear, or you become a department chair, unless you know it all. And so if these people say that something's not worth pursuing, then you don't do it. And then I also had this concept that if you have a really great idea then you present it to the people who are the pros, the people who work in industry, and if the idea really has merit, they're going to run with it, and you're done. And then I've learned over time there may be some flaws in this approach. So we're now in 1978, and this individual who I've told you two stories about too is now a freshman, first-year medical student at Johns Hopkins, and two of his professors share in the Nobel Prize in physiology or medicine. Dan Nathans, who went on to become the president of Johns Hopkins, and Hamilton Smith, his partner. And along with Werner Arber, they shared the Nobel Prize for the discovery of restriction endonucleases. Basically, they created, in so doing, the field of recombinant DNA technology and molecular genetics. And this is a summary of all the patents and licensing fees that accrued to Johns Hopkins as part of this process, which is to say there were none. They did what was the Hopkins approach at that time. If you discovered something, you quickly got it out in the public domain, out in the literature where it would benefit mankind. So the idea of applying for a patent would have been, at that time, repugnant to these brilliant gentlemen. So there's no patents, and Hopkins, to the best of my knowledge, has received, and to the knowledge of our current university president and dean, has not received $1 related to the work that they did. Now, there's a school over here on this coast, UCSF, and one of their professors, Herb Boyer, with some of his colleagues, had a different approach to thinking about patenting and potentially commercializing technologies. And so I have here a photograph of beautiful Genentech Hall on the UCSF campus, funded through funds from Genentech, the company that Herb Boyer co-founded, that went on to do all this amazing work when it comes to translating recombinant DNA technology into therapies that can be used for patients. Around the time, 2006, 2007, a list of organizations with the most U.S. biotech patents in the United States is issued, and you can see up at the top here, here's Genentech in 2007, 2006, University of California, others here, U.S. government and some other organizations, but one organization that you will not see on either list was my university at the time, Johns Hopkins, which was in both of these years, and for a few years before that, the #1 recipient of federal research grants amongst universities in the world. And we don't appear anywhere in the top 10 when it comes to patents. May not have been anywhere in the top 50, simply because the approach at Hopkins was, that's not something we're going to do. So in 2011, the medical student, assistant professor, and associate professor I mentioned to you earlier is now a gray-haired institute director in Baltimore. And he's talking with one of his professors and discussing that it's just too bad there's not a way to deliver controlled drug levels into the eye. I'm explaining how people are getting all these injections into their eyes every month to try to deal with some of these conditions and how that exposes them to increased risks. And we have this discussion and about, well, shouldn't it be possible to create a way to use nanotechnology to deliver drugs into the eye so that it might be possible every 6 months or every 12 months an injection could be done to deliver this drug. So my colleague's professor, his name is Justin Haynes, says, well, let's form a company. And this was a defining moment for yours truly, because I thought, at this point, I've proven to not know how to form companies, and I can't do it. And I said, well, it turned out Justin had done his training up at MIT, and he had formed as a trainee. He'd formed one or two companies already. Two of his companies have FDA-approved products. So he said he would walk us through this process. So we did it. And for me, it was quite an interesting learning process. That's Justin there up on the left. He named this company Graybug. Those are the nicknames of his and his wife's two daughters. And I've noticed with a lot of academicians forming companies, they usually use the names of a loved one, spouse, or children. And this company is based on using nanotechnology to develop products to create sustained release and therapeutic levels within the eye. Now, maybe spend 1 minute talking about the science, but I'm not sure necessarily the science or the need to to outcompete another company is the main reason to do this. One of the key reasons to form a company, I think, is the people that you can work with as part of going down this process. And the people that we got to work with as part of this included Jeff Cleland, who's already been a successful businessman and came on as the CEO. Charles Semba, the Chief Scientific Officer, you may know that name. Charles was the Chief Scientific Officer who drove Sidra to become, through FDA trials and eventually FDA approval for the treatment of dry eye. And then Jerry Cagle, who had a long and distinguished career as the Vice President for Research and Development for Alcon, and fathered through trials and approvals many of the medications and therapies that we currently take advantage or take for granted in treating so many of our patients. So it's possible when you, because as an academician, if you've not been in the world of industry in a significant way, you need people who have been in that world of industry. And there are these really high-quality people that you can get to work with as you do this. So Jerry joined the board of directors of this company, and Charles, as I said, Chief Scientific Officer. And you really work with really very excellent, high-quality people, and they have complementary perspectives and skills to those of us who are full-time academicians. So this drug trial, which is in phase 2, looks at the use of, in this case, a small molecule for the treatment of AMD. Peter Campachero is one of my professors who has a long track record of working with industry for testing potential anti-VEGF therapies in his laser-induced model of choroidal neovascularization. And oftentimes, the responses that he achieves in his model are predictive of the responses that occur in the clinic in patients. So Peter tested this, and it looked very attractive in his model. And this has formulation now of 6 to 12 months of therapeutic levels as a result of these particles. You inject these particles through a very narrow gauge, maybe 30 gauge or a smaller needle. And once inside the eye, they assemble into a large single particle, which then creates a release of drug at a controlled level. So this is still in trials, and maybe it'll succeed, maybe it won't succeed. As you saw this week with Biogen, where billions, tens of billions of dollars of value of the company were lost when they reported a negative trial for their therapy, potential therapy for Alzheimer's disease. This is a high-risk business, and most of these technologies, most of these companies will fail. I think we, as ophthalmologists, we're not into failure, right? I mean, our surgical success rates are quite high, and our ability to help most of our patients is quite good. And so I know I'm uncomfortable. I think many of us are uncomfortable with the idea that we might engage in something where the success rates are low because we hate failure or fear failure. And I think it's okay to fail. Certainly, Thomas Edison would say he never failed. He just found other ways that he couldn't get the light bulb to work, but eventually he would get where he wanted to go. So I think if we overcome that fear of failure and embrace the idea that we perhaps can't rely on others to translate either our clinical observations or our laboratory findings into therapies that might touch patients in their lives, that we can get more involved, directly involved in forming companies and involved in industry. Now, we've continued our work with our nanotechnology people, and one of the discussions that we had, it's interesting, if you take a 10-0 nylon suture and you show it to somebody like me, we would say, well, that's a suture. If you show it to our people in our nanotechnology group, they would say, oh, that's a nanofiber. And they have a lot of interest in work with nanofibers. And so we began to think about, could there be a way that we could create perhaps another generation of sutures that could be available to help patients. Suture technology has not changed all that dramatically for quite a while. There's some evidence that sutures were used thousands of years ago, and ant pincers to use to suture wounds, closed wounds, is something that's probably been around for 1,000 years, and with increasing prevalence of managed care, perhaps will make a comeback before too long. But catgut sutures, 900 AD. Sterilization is only as recent as 1865. The nylon suture, and some 1938. So some 81 years that we've had the suture that we mostly use today, at least we cornea people. And then some synthetic polymers in 1970, and basically nothing since then, although there was some interest in early 2000 about coating a suture, taking it through, giving it a coating that would have some antibacterial properties. So we began to take a little look into what could be done with these fibers, and could there be a way to improve suture technology. It turns out of all the medical devices implanted in people's bodies in the world every year, sutures are by far the most common. 80 million people, about the population of the country of Germany, receive sutures every year. So I have some typical German people here to help set that stage. Now, sutures, we know, are necessary in many cases, but they're also a source of infection. And suture lines and sutures become contaminated. When I was a second-year resident at Wilmer, we always operated with Dr. Malmany, and we were doing extra caps in those days, and a patient came in for just like a 2-week follow-up, and there was a loose suture. And Dr. Malmany said, you have to remove that suture. And I said, but Dr. Malmany, the wound won't be healed already, right? Is that safe to remove that suture at only 2 weeks? And he said, if you don't remove that suture, there's a 100% chance this eye will get infected, so you have to remove that suture. Because bacteria glom on to these sutures. There's a biofilm, they adhere, and sutures become a nidus for infection and infectious organisms. Fairly a simple study where people have looked at removing corneal sutures and culturing them routinely. If the suture is nice and tight and you cut it, presumably it's buried, so on, and you cut it and culture it, about 6% of those will grow organisms. If the suture is loose or broken, almost 40% of them will harbor organisms, and they'd be the ones you'd expect, Staph epi, Staph aureus. So the bacteria really like these sutures, and they do a very nice job of adhering to them. And we thought, well, what if we could make a suture out of antibiotic or certain drugs that we might want to have present at the suture line as the area is healing, and maybe not elsewhere? Why should we have to treat the entire body or use even antibiotic eye drops, which our patients don't always do very successfully, just to get some antibiotic right where we want it, which is right where the incision is. So we looked at seeing if we could develop sutures that contained or were essentially partially comprised of the drugs that we might want to be present in that location. So there's turns out there's a procedure called electrospinning, and you fill up a syringe with medication or the polymer or whatever you want in the suture, and you squeeze that out, and under electric charge it turns into as I said, a nanofiber, or those of us who are used to looking at these and thinking of them as sutures would call a suture. And we also need to get these to form so that they're strong enough so that they have the strength we need to tie and close wounds under the appropriate level of tension. So here's what we found. We found that by playing around a little bit with the composition of these sutures using antibiotic, in this case, these are ofloxacin we've used, off-patent antibiotic, and at increments of 8%, 16%, 24%. Where 24% by weight of the suture is ofloxacin, we can get a 10-0 nylon suture that's has the same breaking strength as the 10-0 nylon sutures that we currently use in our clinics today. We could actually make sutures out of up to 40% of antibiotics. So we're getting where it's getting pretty close to the suture is crystalline antibiotic. But as we go beyond 24%, we start seeing that the sutures become easier and easier to break. These sutures, depending on how we make them, can release, in this case, antibiotic, in this case, ofloxacin, for weeks after they're implanted or placed in saline. And here in these two culture dishes, this is one of these nanofibers with no antibiotic in it, and you can see a uniform layer of organisms. And here there's a zone of inhibition around this suture due to the release of the ofloxacin. And as those of us who are fortunate enough to be ophthalmologists know, even though there's, quote, increasing resistance of organisms when tested using standard microbiologic analyses in microbiology laboratories where the levels are in the microgram per ml or so concentration, we can get effective killing using these sutures, even with organisms that are, quote, strongly resistant to the antibiotic. So the concept here is that it would be possible, depending on the procedure that we are performing and the type of wound healing modification or prevention of infection that we seek, we can create sutures, say for phacos, that contain antibiotics, steroids, or non-steroidals. After trabeculectomy, where we want to control wound healing, we might perhaps want to include agents that alter the wound healing response of the local tissue and avoid potentially the complications by allowing the drug to go to other places in the eye. And maybe after keratoplasty, where we want to have a long-term effect of reducing risk of graft rejection, we could use the appropriate agents that might reduce that risk. So a few thoughts that I have about why should those of us who perhaps did not grow up working in industry, why should we consider forming companies? One is, I think that you clinicians understand best what are the needs of patients and what are the unmet needs. I remember one of my first year residents who is a guy named David Geyer, who went on to form the first company with the first treatment called Macugen for treating macular degeneration. And he said he had the idea for the drug, and he went around and talked to many of the large pharmaceutical companies, the names of which you know, and he would sit in a room with the business development people, and they would tell him that there was no market for a drug to treat AMD, that even if patients went blind in one eye, they still had another eye, and they just had zero interest in pursuing this. And he felt basically forced to create his own company. And the rest is history. He showed there was a market, and the big players really did get involved in it. So I think it's we clinicians who best understand what are the unmet needs of our patients. I think for a product to reach patients, we have to understand that there has to be that intellectual property protection. And that's something that even at Johns Hopkins, there's been a dramatic change in our understanding of that. Our university president blames the faculty for some of the economic challenges faced by the city of Baltimore. And he says, look at what a great job they've done up there in Boston and Palo Alto, starting companies, creating jobs for people. It's you professors' fault that there aren't these companies and so on done here. And that had a big impact on people. They paid attention to that argument. But without intellectual property and patent protection, there is no economic path forward. I think large companies just aren't necessarily well prepared to understand the needs. They think of some products that are significant in ophthalmology as being too small to move their needle, and so they are not going to pay a lot of attention to it. Or they need, there's a lot of pressure on these pharmaceutical companies by shareholder activists, et cetera, to develop revenues in a year or in 2 years or 3 years, but not in a decade. So the small company that you might help form, really focused in this area, can make a major contribution by de-risking the asset, by basically showing up to phase 2 trials that this has a very good shot at being effective. And then the big pharmaceutical companies then will swoop in, and because they have to, they will pay handsomely as a result for that de-risked asset. Department chairs, and this will come as a shock to a lot of you in the room and will tell me that I don't know what I'm talking about, but department chairs do not know everything. And so if you get a review back from leaders in the field, including department chairs, where they say your idea has no merit, don't put too much store by that. Don't accept that at face value. I'm sure they're doing their best, but they're all products of their own upbringing as well, those department chairs. And if it were easy, they'd be doing it already. And in many cases, forming a company may be the only way that your insight will ever be able to reach patients and improve their lives, because you can't necessarily rely on the professionals, industry, and those players to realize that something that's going on in your laboratory has this tremendous potential to either expand or create a whole new market. So I think it's those people, the clinicians, and I heard a lot of really good presentations already this morning on potential new therapies to touch what might be relatively orphan indications, but really drive forward the field. And I think it's going to be those of us in this room with MDs that can best appreciate what our patients need and hopefully either make the case to industry or become industry, at least part-time, and go through this experience. And it's fun. And it's a way to learn a whole group of new things and work with new people that you might not otherwise get to work with. So I would strongly encourage, if I were an assistant professor today, I would be thinking in terms of not if, but when I will get involved and when I will start my first company, and not feel ashamed if it's not successful, because we're going to learn one way or other. We're going to learn a tremendous amount every additional company that's started, including those things that aren't successful. And so I wish I'd had When I was a young assistant professor, somebody would take me aside and say, it's okay to think about creating products that touch people's lives. It's not unethical, it's not wrong for a physician to think that way and to try to develop products and to patent technologies and develop intellectual property. So it's a real treasure and delight to be asked to be able to give this lecture in honor of Stephen Foster, who's contributed so much. And as I said, in my view, maybe his major contribution has been all the bright young people that he's populated around this country and the world, and will carry on his work many decades into the future. So thank you very much.
Anybody with a question or comment?
So, Dr. McDonnell, when you talk about the pockets of hope, I romanticize that era. Where it was science for the sake of science, it wasn't about a commercial or end goal. And I guess when I look at modern society now, in modern biomedical society, there are people from the 60s who, went on to become chairman and whatnot from the NIH, and they look at today's science and things that get patented all the time, and we would never patent this kind of idea back in the day. And I think we're kind of getting to this wall of sorts where, in the rush to commercialize everything, we're creating a gridlock for innovation because there's so many patents for so many things. There are broad patent languages, and I feel like that's kind of the flip side of your coin. I actually think we need a little more of those purist, romanticized views of science, because I think we're getting a little too commercialized in some ways for the good of society.
Well, I think you make a good point. I think we see in technology fields, all the big companies, Apple, and everybody's involved constantly in lawsuits over intellectual property with our smartphones, et cetera. And people are trying to streamline how patent disputes are handled and reduce the cost and the effort to do that. And I would agree with you. I think that there needs to be a balance. Wilmer faculty in the last, 4 years have founded 8 companies. And I think with the understanding at Wilmer that there is, it's not instead of your NEI funding or your NSF funding or wherever your current hypothesis-based funding comes from, but it would be in addition. And I hope that what we'll see is a reasonable balance, and for most people that they can strike the balance between becoming a patent troll and just trying to patent and control everything to not add value, but to extract value from successful companies. And I hope we can get there as a society, but I think that, and I don't want to pick on anybody because they were exemplary professors and exemplary scientists, but Hamilton Smith and Dan Nathans to patent nothing is probably, I think, too far at that extreme. And in theory, Baltimore could have been the epicenter of the biotech industry, and obviously it's not. Baltimore has a different reputation, beautiful as it may be. And we look forward to having everybody at ARVO in Baltimore in a little over a year. But we potentially lost an opportunity there.
Peter, thank you so much.
I just wanted to make a comment. I thought that was a fabulous talk. I've sort of lived through this myself. I trained with Dr. Foster and Kwan 25 years ago and sort of rode the wave of anti-VEGF innovation. I was on the original Lucentis trials, and it was really a blast. But at the end of 20 years as a retina specialist, I found myself stagnating a bit. And then I went back, got an MBA, and had an opportunity to work with Dave Geyer at Optotech, and just recently helped launch Luxturna at Spark. And I can tell you, it's been a blast. And I couldn't agree more because with your thesis, because basically academia comes up with the ideas, industry funds those ideas in the trials, and then practitioners do the trials. And only if those three sectors collaborate, can we get innovation? And I think it's really great to hear someone from Hopkins espouse those ideas.
Well, the thing about Hopkins may take us 50 years, but eventually we'll do the right thing after exhausting every other possibility. Is what so I think people at some other institutions laugh at. All of a sudden, we've figured out that maybe this is okay to do what they've been, setting this standard for decades. But I'll admit we were late to the party, but hopefully now that we're in the party, we can contribute in some significant ways. And I'm very proud of my young faculty that are doing these, assistant professors, associate professors, starting companies, because I think to have that experience so early in career is really going to be helpful. And it helps people to understand better, appreciate. One of the things I think that my scientists thought was that they love science for science sake, and scientists in industry love science to make money. And what they've learned by working more with industry scientists is the love of science is shared, and industry scientists can really be complementary to the full-time academicians. So I think they've really come to enjoy the experience and break down the barriers between the halls of academia and industry.
Yes, I was just curious as to the industry. Well, when you're a faculty member form a company, I'm assuming the patent was filed by Hopkins, and Hopkins has a certain percentage on that. And then, so how does the percentage in the relationship with these companies that are spun out of the university, there's a relationship there. What is that financial relationship?
Yeah, and all this that I mentioned presupposes, people are very upfront and candid with the conflict of interest, conflict of commitment committees, and make it all very clear. And the way it works at Hopkins is you've come up with something that you feel is a great thing, and the thing about academicians, at least at my institution, is often everything they do they're in love with and they think is great, and so and should be patentable and so on. But the university will hopefully validate that assessment, pay for the costs of securing the intellectual property, and then as a department chair, I am agnostic as to how it gets to patients. Maybe Merck or Pfizer or somebody comes by and finds out about it and says, oh, this is great, we'll license it at this very early stage and we'll run with it. Or the person may say, well, I want to form a company around this. And the way it works with the royalty distributions is that the university gets a third, the inventors get a third, and then the School of Medicine, the dean, and the department, me, split the other third. And the third that comes to my department, half of it comes to me, and the other half goes to the inventor's laboratory. So the inventor gets another source of funds that go through it. And so it may or may not be a life-changing event. In most cases, it's not a life-changing event for people, but it's nice, and we probably have now maybe 30 ongoing licensing fees that come in every quarter. I get a little statement, and the numbers, I hope, will grow over time. But it's sharing, and but the inventors, it's a third, and the university does, I think, pretty well in covering its costs, certainly, of its tech transfer office through that. But I think our numbers are much smaller than, say, MIT's and Stanford's numbers. Great, thank you.
So it's always in appreciation for your contribution, Peter. We have created a slide that David's going to read for you because it's better eyesight than me. And go ahead.
You want me to read this?
Yeah, sure. To read out. So,
okay, 2019 Sonoma Eye in Art is honored to present to Peter J. McDonnell, MD, William Holland Wilmer, Professor of Ophthalmology, Director, the Wilmer Eye Institute, the Johns Hopkins University School of Medicine, with deep appreciation for your distinguished contribution as the second annual C. Stephen Foster, MD lecturer.